Assalamualaikum warahmatullahi wabarakatuh and hello everybody. In video 3 of genetic inheritance, you will learn about Mendels First Law, Law of the Segregation, and Mendels Second Law, Law of Independent Assortment. With me, Puan Rosmariza binti Muhammad Alaya Syafi'i. Learning outcomes. At the end of the lesson, you should be able to number 1, state Mendels First Law, Law of the Segregation. Number 2, state Mendels Second Law, Law of Independent Assortment. Mendels First Law, Law of the Segregation. Law of the Segregation. This law derived from monohybrid inheritance, inheritance of a single characteristic. For this breeding experiment, Mendel chose varieties that differed in only one character, such as color of the flower, could be either purple or white flower. It all started with Mendel's pea plant experiment. Mendel crossed the true-bred purple flower with true-bred white flower. For each true-bred plant of the parental generation, they has two identical alleles, denoted as either homozygous dominant or homozygous recessive. Mendel controlled the pollination by removing stamens from the purple flower, and then he transferred the pollen from the stamens of the white flower to the carpel of the purple flower. Pollinated carpel matured into pod. Mendel planted the seed from the pod to form F1 generation. Mendel found that the first offspring generation all has purple flower. Every time he repeat the experiment, he always saw that the F1 generation is always produce purple flower. He also tried this experiment with other traits and still he got the same result, that the F1 generation offspring always resemble one of the dominant parent and never the other parent. Because of this, what he did that he made a cross-pollination or self-pollination of the F1 generation to produce F2 generation. And what he observed that almost 75% of the F2 offspring were produced purple flower. The remaining 25% of the F2 offspring produced white flower. The recessive trait of the parent will only reappear in the F2 generation. It means that all this time, the F1 generation actually carries the white alleles, but it was actually being hidden or not being expressed. So, Mendel proposed that for each trait, plant contain two heritable factors, later on known as a gene. The idea that two heritable factors segregate from each other during gamete formation is known as Law of the Segregation. Law of the Segregation states that two alleles for a heritable character will segregate or separate from each other during gamete formation and end up with different gametes. You guys have to memorize this law. To demonstrate what is meant by this law, let's take a look at this diagram. We have homozygous dominant parent number 1 and homozygous recessive for parent number 2. Before undergo meiosis, each heritable factors must undergo DNA replication. Each chromosome replicated to form sister chromatid. So, at a metaphase 1, we have a pair of sister chromatid, or also known as homologous chromosome. In meiosis 1, homologous chromosome will separate and each sister chromatid will end up in 2 different daughter cells. In meiosis 2, the sister chromatid will split at the centromere to allow the daughter chromosome to migrate and end up in the different gametes. This is where we have to discuss about the law of the segregation. Remember, law of the segregation states that 2 alleles for heritable characters will segregate or separate from each other during gamete formation and end up with different gametes. Mendel's Second Law, Law of Independent Assortment. Law of Independent Assortment derived from dihybrid inheritance. Dihybrid inheritance means that inheritance of 2 characteristics which controlled by a different genes at the different locus on different chromosome. For this breeding experiment, Mendel chose varieties that differ in 2 characters. For example, seed color could be either yellow or green color. Seed shape could be either round or wrinkled shape. Law of independent assortment states that each pair allele segregates independently of any other pairs of allele during gamete formation. To demonstrate what is meant by this law, let's take a look at this diagram. We have heterozygous parent. Before undergo meiosis, each gene or heritable factors must undergo DNA replication during S phase. Each chromosome replicated to form sister chromatid. So, at metaphase 1, we have 2 pairs of sister chromatid, or each we call homologous chromosome. This is where we have to discuss about the law of independent assortment. Remember, law of independent assortment states that each pairs of allele segregate independently of any other pairs of alleles during gamete formation. These 2 will separate independently of these 2, and likewise. It means that within the cell, these 2 can be arranged like this, or they can actually be switched like this. Okay, focus on the chromosome here. For the second gene, on this side, we have dominant allele, and this side we have recessive allele. And since this allele independent from the first allele, the position can be switched into like this. recessive allele at this side and dominant allele of this side. Because of this independent assortment, half of the cell will have this arrangement and other half of the cell will have this arrangement. The cell then undergo meiosis 1. Homologous chromosome will separate and the chromosome at this side will migrate to the daughter cell. The chromosome at this side will migrate to this one, daughter cell. Same with this one. In meiosis 2, both sister chromatid will split at the centromere to allow the daughter chromosome to migrate and end up into a different gametes. Okay, thank you. That's all for today. See you next time.